Hello everyone. The subject of today's lecture is from Chapter 12 in Essential Biochemistry, and we were going to go over an overview of metabolism and free energy. So some of this is review from general chemistry. First off, when we say metabolism in biochemistry, we refer to the network of chemical reactions carried out by living cells. So this is where the biochemistry comes in. We have the chemical reactions, which are the chemistry, and the living cells, that's the biology. And metabolism consists of both catabolism and anabolism. So catabolism over here on the left is when macromolecules from our diet are broken down, and when they do so, they release free energy, which is captured. They also release building blocks, smaller molecules, and then these building blocks are then, along with the captured free energy, are used in anabolic process to build up the macromolecules that make up our bodies. So we really are what we eat. The food that we eat is broken down into the building blocks that make the different structures of our bodies. So these pathways are all connected. The pathways produce specific products. We can refer to reactants, intermediates, products as a general term called metabolites. So I have this from the Moran textbook. This is an overview of catabolism, and when in the future I give you pathways, I'll have a sort of like a where are we place where I will show you where we are, because there are lots of pathways that have lots of steps, and sometimes it's easy to get lost. My anabolism slide looks like this. So I'll be sure to point out when we're talking about a catabolic pathway or an anabolic pathway. So first, let's start with catabolism and just talk about some general principles. We have our large nutrient molecules. So our diets consist of proteins, polysaccharides, and triacylglycerols, nucleic acids, not so much. But they're broken down into their constituent parts, proteins to amino acids, polysaccharides to monosaccharides, and triacylglycerols to glycerol and fatty acids. And these are broken down into 2 and 3 carbon intermediates. So the 2 carbon intermediate is acetyl-CoA, and the 3 carbon intermediate is pyruvate. And these nutrient molecules are oxidized when they go from their monomeric units to the 2 or 3 carbon intermediates. They can be further oxidized in the citric acid cycle, which completely oxidizes those carbons to carbon dioxide. And in so doing, donates protons and electrons to NAD plus and Q. These are coenzymes to form the reduced NADH and QH2. They then donate the protons and electrons to oxygen to form water. This is a highly exergonic process. This is down here at the bottom. And some of the energy is captured to phosphorylate ADP to make ATP in a process called oxidative phosphorylation. So I'll be referring to this pathway as well. So in anabolic pathways, these are biosynthetic pathways. We start with the small metabolites that are the building blocks that our food is broken down into. So those are the starting materials, and they're oxidized. So when we use those to make larger biomolecules, the larger biomolecules are reduced. So that means we need a source of protons and electrons. And here the coenzyme involved is NADPH. So that serves as the proton and electron donor, and in the process, it becomes oxidized to NADP+. So these reactions are endergonic, or they would be, unless we added some free energy. So that means that these reactions also require ATP, that captured energy from the catabolic pathway. So we have to couple these endergonic pathways with the exergonic hydrolysis of ATP in order to get the entire overall process to be exergonic so that it will occur. Catabolic pathways involve degradation, so we take our larger reduced biomolecules from our food, oxidize them to smaller metabolites. So in oxidation, protons and electrons are given up. They are given to largely to NAD+, which serves as the electron acceptor, and in so doing it becomes NADH. These reactions are exergonic. They release free energy, and because they release free energy, we can use some of that free energy to produce ATP. And then that ATP will be used to drive the anabolic reactions that I described on that previous slide. So before we go on, we want to be sure that we define and describe three important thermodynamic terms: the delta G of reaction, the delta G zero prime of reaction, and the equilibrium constant. So let's start with the delta G of the reaction. Well, this comes from the Gibbs free energy equation. So G is Gibbs, so this is free energy. Delta G equals delta H minus T delta S. So free energy has both an enthalpy and an entropy component, with enthalpy is delta H and entropy is delta S. The T is in Kelvin. And I like to define this as the amount of free energy available to accomplish the reaction. So that's the term I'm going to be using. And it does combine the enthalpy, the delta H, and the entropy, the delta S terms. And we can use the value of delta G to figure out if the reaction is spontaneous. So the reaction is spontaneous if the delta G is negative. So if the delta G is negative, that means the reaction will occur spontaneously and no input of energy from the surroundings is needed. Another way of saying that is there's free energy available to accomplish the reaction. And the term that we use here is exergonic. So a reaction with a negative delta G is exergonic. If delta G is positive, the reaction won't occur spontaneously. Another way of saying it, that is that there's no free energy available to accomplish the reaction. So if that's a reaction you want to occur, then you'll have to couple that with another reaction that provides an input of energy from the surroundings. By itself, the reaction is endergonic if the delta G is positive. So how do we know if we have a negative value? This chart is nice because it shows that reactions are favored. You'll get a delta G that's negative. You can see mathematically if the delta H is negative, that's an exothermic reaction. If delta S is positive, that means the products are more disordered than the reactants. Here we have an exothermic reaction with a reaction that produces products that are more ordered. So if you do the math, that will occur spontaneously, but only below a temperature of delta H over delta S. Here we have an endothermic reaction, but the products are more disordered, so that's spontaneous at temperatures above T equals delta H over delta S. And if we had a reaction that was endothermic and become more ordered, that is not going to be spontaneous ever at any temperature. So the information given by the delta G is that if the delta G is negative, that means there's free energy available to accomplish the reaction at the beginning. Now, as the reaction progresses, the delta G becomes less and less negative until delta G reaches zero, and then that will be at equilibrium. And where equilibrium is positioned depends on the reaction. Some reactions go all the way from reactants to products, and some only go part way. It just depends on the reaction. What we're not given is path and rate. We don't know what path the reaction takes, and we don't know how fast it takes to get there. The quintessential example of a delta G reaction that's negative is rusting, like a bridge that rusts. It's spontaneous, but it's very slow. Okay, so let's look at the delta G zero primed of a reaction. That's a notation that looks similar, but it's very different in terms of how we define it thermodynamically. Each compound that we are familiar with has its own delta G of formation, and that corresponds to the compound's free energy in its standard state. So you might remember standard states from general chemistry, where we defined the temperature was 25 degrees Celsius, which corresponds to 98 Kelvin, 1 atmosphere pressure of your gas, 1 molar concentration of your solution, and the water is left out because water has a constant sort of concentration. That is, it just has the constant number of moles in a liter. So there are 55.5 moles of water in a liter of water. But biochemists add something else because our reactions don't occur at an H plus concentration of 1 molar. So we're going to make an exception to the reactant concentration, and we're going to call that the H plus concentration is 10 to the minus 7th molar. So those are biochemical standard states. So what we do is we add an apostrophe onto the delta G zero prime. So we add an apostrophe and we pronounce that primed, and we indicate that the pH is 7, which corresponds to H plus concentration of 1 times 10 to the minus 7th molar. So a delta G zero primed reaction is the sum of the delta G of formation of all the products added together. That's this big sigma there. That means sum. Subtract from that the sum of the delta G of formation of all of the reactants. And the difference is the delta G zero primed of the reaction. So what that means is that every reaction has its own delta G zero primed and is a constant. The value is negative if the reactants have a greater delta G of formation, and the value is positive if the products have a greater delta G of formation. So that's the two delta G's. How about the equilibrium constant? Well, here you might remember from general chemistry, we look at our balanced reaction, and we take the molar concentration of the products divided by the molar concentration of the reactants. If there were a coefficient here, we would raise the concentration to the power of that coefficient. So what happens to A, B, C, and D in this reaction as the reaction progresses, we can use this equilibrium constant expression and call it KEQ if we're at equilibrium, but we can also use it to describe the ratio of products to reactants at any point along the reaction. And if we're not at equilibrium, we have to call it something else. We call it Q. So what happens as the reaction progresses, assuming that it's spontaneous, that the concentrations in A and B will be smaller as the reactants disappear, and the concentrations of C and D will increase, and Q will continually increase until you get to equilibrium, and then that's where Q equals KEQ. So these terms are all related. So delta G, let's go back. The delta G changes throughout the reaction, and it indicates the free energy available to accomplish the reaction. The delta G zero primed is a constant, and it looks at the differences of the delta G of formation between the products and the reactants. R is the gas law constant, T is temperature in Kelvin, and this is the ratio of products to reactants. So I wanted to look at, this is an old textbook, as you can see that there's a kilocalories per mole here. This looks at a hypothetical reaction where you take reactants A plus B and you get a product AB. And suppose this reaction looks like this at the end, where you have lots and lots of AB and almost all of the A and the B get together to form AB at the end. So this is what it would look like at the end. Lots of reactant in the beginning, almost all of the reactant gets transformed into product. So depending how far you go, you're going to get K values from 1 all the way up, say, to 10 to the 11th, depending on how far you go. If you're way up here at 10 to the 11th, you have virtually no reactants left over at all. And how that corresponds to delta G is the more the reactants get transformed into product, the greater the negative value of the delta G. Now, suppose I didn't have this in my book, so I had to make my own table. Suppose I have a reaction that's not favored. So here's my reactant, and very, very little of it at the end is transformed to product. So here we have very, very little product, and we still have lots and lots of reactants. And here our delta G zero primed are going to be very positive values. Say if we have something very small like this, virtually none of the reactant is transformed into product. So it just depends on the reaction. We can have all of these values of an equilibrium constant from, say, from a negative 65 all the way through positive 65 and everywhere in between for our delta G, or our K values from 10 to the 11th all the way to 10 to the minus 11th and everything in between. Again, it depends on the reaction. So from the delta G, remember I said delta G changes. The delta G value tells us if there's free energy available to accomplish the reaction, and that occurs when delta G is negative at the beginning of the reaction. As the reaction progresses, and it's spontaneous because the delta G is negative, the delta G becomes less and less negative as the reaction progresses until it gets to be 0. And when delta G is 0, the reaction stops. Well, it doesn't really stop. It just looks like to our eyes that it stops. But the reaction is equilibrium. And remember, we defined that as the rate of the forward and the reverse reactions are the same. So at the beginning of a reaction, assuming it's exergonic and spontaneous, and we have a large negative delta G, there's free energy available to accomplish the reaction will go. And this delta G is 0 primed is a constant. It just tells us the difference in the delta G of formation between the products and the reactants. And at the beginning, we have lots and lots of A and B, but not a lot of C and D, because C and D are the products. So that means this overall ratio is going to be, Q is going to be small, because we have lots and lots of A and B and not much C and D at the beginning of the reaction. But as the reaction progresses, the delta G will become less negative. This delta G 0 primed is a constant, it doesn't change. But A and B are going to disappear, and C and D are going to appear, so this value of Q is going to get larger until we get to equilibrium, where our delta G is 0, and there's no more free energy available to accomplish the reaction, so the reaction stops, or it appears to stop to our eyes. It's really the rate of the forward and the reverse reactions are the same. That's a constant. And now we have our Q, and it's equal to the equilibrium constant, because now we're at equilibrium. And again, this KEQ, that just depends on the reaction, what it happens to be. I just, in that table, gave you a whole range of the possible KEQs you can have for all the reactions in the world. So if this is true at equilibrium, I'll go back here, if delta G is zero, I can write an equation that describes the relationship between the equilibrium constant and this delta G 0 primed. Now I want to say you can only use this equation at equilibrium, and the two reasons why are delta G, not delta G 0 primed, delta G is 0 at equilibrium. And I didn't say Q here, I said the equilibrium constant KEQ. So that means if you use this equation, you better be sure you're at equilibrium and you're not somewhere at the beginning or in the middle or somewhere along the way. So the criterion for spontaneity, if we're at the beginning of a reaction, is that delta G is negative, the reaction is spontaneous. We don't use the delta G 0 prime because that's a constant. We could look at Q. So if Q is smaller than the equilibrium constant, then Q will increase its value by decreasing A and B, the reactants, and increasing C and D, the products, so that Q is equal to the equilibrium constant. Okay, so now I want to sort of segue into the two types of metabolic reactions, near equilibrium and metabolically irreversible reactions, because we're going to use these values of delta G and KEQ to figure out which is which. So near equilibrium reactions, the Q is at the beginning of this reaction is pretty close to K. So that means you can reverse the reaction fairly easily just by adjusting the reactants and products concentration. So here, if the reactants are at high concentration, you'll go in the direction towards products. But if the products are at high concentration, you can easily reverse the reaction because Q is already pretty close to K. If you have a good biochemistry textbook, you should be able to figure this out because it will show the reaction with equilibrium arrows, where it will show the reactions are reversible and you've got two arrows, one going in each direction. The enzymes can act quickly in the appropriate direction to restore equilibrium. The reason we're interested in these reactions as biochemists is because when we talk about our pathways, we want to talk about how the pathways are regulated. And these enzymes do not regulate the pathway. So we'll be looking for those double arrows to show we are going to regulate the pathway, but not here. Metabolically irreversible reactions, Q is far from K, and delta G is a really large negative number. And what that means is essentially all substrates converted to product. And that means that it's not going to be feasible for us to change the direction, no matter how much we increase the concentration of the products. We can't get it to go in reverse. In a pathway diagram, the artist should show you these reactions with an arrow in only one direction. So it should be easiest for us to spot this if the artist has done their job. We're interested in those because these are the key regulatory steps, or the committed reactions in a pathway. Here, the enzymes saturate with substrate, so we can't regulate the activity of this enzyme by with the concentration of the substrate. The only way is to use allosteric modulators to turn the enzyme on or off. So let's see how we can do that. But first, let me show you that if we go through a pathway, the flux through a pathway is constant, and it is set by the rate determining or the slowest step. So that's where we're going to control a pathway. So we can either activate that enzyme or speed it up, or we can slow it down. We should be able to spot this in a pathway diagram because it will show the arrow in one direction, or we could look at the delta G, and if the delta G is a really high negative number, we know that their reaction is only going to go in one direction. So what that means is that these catabolic and anabolic pathways I was telling you at the beginning, they're not the same because both pathways have to be exergonic. So if you have a catabolic pathway that's exergonic, you can't just reverse it and have an anabolic pathway, because if you reversed it, then it would be endergonic and then it wouldn't happen. So the anabolic pathways have to be different because they have to be exergonic too. So how are we going to control this? Well, there are lots of ways the cell can do this. So one is by allosteric control, which I'll describe to you. The other is covalent modification, and I'll describe that as well. We'll talk less in this class about genetic control because that's covered in other courses, but here you can control this at the synthesis of the enzyme. So either at the level of transcription or translation, rather than regulating an enzyme that's already around, the cell can decide, well, I'm going to make that enzyme or I'm not. I'm going to transcribe and then translate this protein and make enzyme, and that way the pathway is active, or I'm just not going to make the enzyme, and if you don't make it, then the pathway will be off. That's more of a long-term control. So allosteric control is regulating an enzyme that's already there, and this is short-term. And there are substrates or products, these metabolites, that serve as either activators or inhibitors that either increase or decrease the activity of the enzyme respectively, that catalyzes this rate determining step or this committed step, the step that commits you to continuing all the way through the pathway. So I have a schematic diagram here. Say I have this pathway, A goes to B, B goes to C, C goes to D, D goes to E, and my eventual goal is to get product P. And every single one of these reactions is catalyzed by an enzyme. Well, say I get to the point where product P piles up and I don't need any product P anymore. So what happens then is P can actually act as an allosteric inhibitor of E1, the committed reaction or the first reaction. So the way that this could happen is that this pink hexagon right over here is an allosteric inhibitor. That's product P. And this diagram right here is meant to represent E1 in the T form. So I have a dimer of my E1, and what I'm circling here with my mouse right here, those are the active sites. So this is in the T form, and remember the T form, the enzyme has a low affinity for its substrate being A. So that's what we want. If we want to shut down the production of product P, we want to make sure that the first enzyme in the pathway is inactive, so we essentially shut down the whole pathway. So in this scenario, E1 is one of those metabolic irreversible reactions. Its delta G value is going to be very high. What about the opposite? What if we really desperately need product P and this pathway needs to crank out product P? And this last step is E4. We want to make sure that if we have this high flux of A, B, C, and D going through the pathway, that it doesn't get to a bottleneck at E4, which is a later step at the pathway. So metabolite B can serve as an allosteric activator for E4. So here I have this is meant to represent E4 as a dimer, and metabolite B is this green triangle. So B is sort of essentially going forward to E4 and say you better be ready because a whole lot of D is coming your way and you need to be active. So this stabilizes the R form, and again I'm circling with my mouse here the active site. So this is where the substrate for E4, which would be D. This is where D would bind. And again, in this scenario, E4 also has a high negative delta G. So this is how I would correctly draw this pathway, where A goes to B, and I only have an arrow in one direction because E1 can be allosterically inhibited by product P. E2 and E3 and E5, those are all reversible, but E4 is not. And E4 is another place where I regulate the pathway where the E4 can be stimulated by metabolite B. So that's a way of carrying out allosteric control. So what do we mean by covalent modification? Well, that means we can attach a phosphate group to an enzyme, and which may turn on or turn off the enzyme. And that's what makes it a little complicated because it depends on the situation. So if you look over here on the left, I have an enzyme. So this is an enzyme with an OH group, and hopefully you remember that OH is amino acid side chains serine and threonine, and also tyrosine. Again, depends on the enzyme. Have this OH group, and this is a good place to covalently attach a phosphate. And we will need an enzyme, a kinase. A kinase will catalyze the transformation of this to a phosphate, a phosphorylated side chain. And one of the phosphates at ATP will serve as the phosphate donor. So kinases carry out phosphorylations using ATP as the phosphate donor. Here, the artist has envisioned that this enzyme is inactive, but you can activate it by attaching a phosphate. Now, we want to make sure this is reversible. We have another class of enzymes called phosphatases, and they carry out hydrolysis reactions. So we have water here. Well, they'll do a hydrolysis and then return the enzyme to its inactive, non-phosphorylated form. The problem is it doesn't always work that way. So I actually redid this slide here. So if I do this, sometimes the way that it works is that, if I go back here, notice that the unphosphorylated form was inactive. Sometimes it works out where the non-phosphorylated form is inactive and you phosphorylate the enzyme, and that inactivates it. So that means that we have to be really careful in figuring out, well, in what scenario does it make sense for me to have an enzyme in the phosphorylated form to be inactive, or what does it make sense for it to be active? And I'll be teaching you strategies where we can figure this out and not have to memorize it. That is the first part of Chapter 12. Thank you very much for your attention and your participation, and I will see you in class.